Hello, my name is Michelle Hughes, and today is Sunday, September 6th, 2020, and I'm going to be talking about our Unit 2 case study, Case Connections. And the two cases that I chose to discuss were number 2 and number 4. So I'm going to start off with number 2, which is a case about the uptake of glucose and in terms of normal cells and tumor cells, and um, basically what modifications have occurred in cancer cells versus normal cells. So to start off, I'm going to talk about how glucose uptake is regulated in the liver, skeletal muscle, brain, and adipose tissues. And then I'll compare that to the tumor cells and talk about the brain scans, brain scan images provided. So in normal cells, most energy is created via mitochondrial cellular respiration because it is an efficient way of generating energy. In the fed state, the liver, skeletal muscle, brain, and adipose tissue all use dietary glucose for the main fuel source. The mechanism of glucose uptake, however, differs. In the liver, GLUT2 is used. In the skeletal muscle and adipose tissue, GLUT4 is used. And in the brain, GLUT1 and GLUT3 are used. And out of those, the brain and the liver are insulin independent, but the other tissues are insulin dependent. So the brain and the liver can produce energy without being fed constantly, but the others cannot. So because the liver can produce glucose even in a fasted state, it's able to provide red blood cells and the brain with a constant source of energy in order to maintain like basic vital functions and homeostasis. In contrast to normal cells, however, cancer cells almost exclusively use aerobic glycolysis. This is much more inefficient than cellular respiration that normal cells use. And I thought this was odd. It kind of seems like a contradiction. Why would it choose to go the more inefficient route? This is known as the Warburg Principle, which I learned about. And it basically states that even though it's more inefficient for producing energy, the rate of uptake exponentially increases. So it can consume a lot more glucose in a shorter amount of time. And it can occur up to 100 times faster than normal cells. So this would be advantageous because the cancer cells are needing a lot more energy. They're trying to get as much energy in as fast as possible. So that's probably one of the main reasons it chooses this form. So in the photo provided, the patient is injected with basically a radioactive form of glucose, and it has two images in the case study. And the one on the right hand side is the PET scan, and you can see pretty distinctly the areas of the tumor and the cancer cells because the glucose is drawn like a magnet to the cancer because of the cancer's high energy demand. So all of the fluorescent glucose illuminates where the cancer cells are in the brain. So that was pretty interesting to look at. Normal cells use oxidative phosphorylation, but somewhere along the line, the cancer cell's metabolism mutated and switches to aerobic glycolysis instead, increasing the rate of glucose uptake. And then I do list 3 references underneath. The next case is case number 4, and this is about a 2-month-old female who's presenting with jittery spells several hours after breastfeeding, and she hasn't eaten anything other than breast milk since she was born. And the case study gives us 4 potential diagnoses, so I'm going to go over those one by one with you. So the first one we have is hereditary fructose intolerance. And this is the inability of an individual to break down fructose. Some symptoms include nausea, vomiting, jaundice, and enlarged liver. And although the patient does have an enlarged liver, that information was given to us in the case study, the child has not had anything other than breast milk to eat in her entire life. And breast milk does not contain fructose. So this can be ruled out. Fructose is normally found in fruits and things like that. So next we have galactosemia. This is the inability of the body to convert galactose into energy. And the main symptoms include jaundice, lethargy, feeding difficulties. And although the patient is showing lethargy, the symptoms for this typically appear within a few days after the infant is born. And because our infant or our patient just started exhibiting symptoms around the 2-month mark, this made me think that this was not the right diagnosis. A third option was glucose 6 phosphate dehydrogenase deficiency. One factor about this genetic disorder worth mentioning is that it's almost exclusively found in males, not females. So that was my first red flag. One of the most common symptoms associated with this deficiency is jaundice and anemia. And basically, the red blood cells are destroyed faster than the body can replace them. So the amount of red blood cells is less than what the body requires. So because that didn't really sound like it fit the description and the symptoms didn't match up, I moved on to the fourth option, which I thought was the closest and could be the correct diagnoses. And that is glycogen storage disease. And this is where basically the liver can't release the stored glycogen into the body for energy use. So what happens when we eat is glucose is released into the bloodstream, and all the cells have the ability to get their energy needs met. And when we have enough energy, the liver stores glucose for future use in the form of glycogen. And when the body is in a fasting state, it releases the glycogen, which is converted back into glucose. One of the most common symptoms associated with this disease is an enlarged liver and hypoglycemia. So both of those align with the patient's symptoms. And other common symptoms that our patient also exhibited were fatigue and seizures. So fatigue, aka lethargy, and seizures, both of those the patient has. And another indicator that this is the correct diagnosis is that in the case study it mentions the patient was given epinephrine to see how her body would react to it, how her blood levels would react. And what we found was that the blood glucose levels did not change, but it did increase the lactate levels. And this is pretty unusual because when the body is introduced to epinephrine, it is a major signal that the liver can release glycogen and break it down into glucose. And so this made me think that there is a malfunction in the breakdown from glycogen to glucose. Another problem with storing too much glycogen is that it can cause the blood to become acidic, kind of toxic. So I think this is the correct diagnosis for our patient. And one of the main treatment options for this disease is dietary restrictions. So so those are my 2 case studies, and I do list the references on the Word doc that I will be uploading. Thank you so much.